Well, welcome to the second lecture of Bio 110. This is going to be the second lecture in signal transduction, and I think that we have enough material here to work on Wednesday and Friday. So I'm looking forward to doing that, and then I think for the other lecture we can work next week on Wednesday and Friday as well. If you didn't hear it this morning, and I forgot to mention it to the morning class, I think that some of you want to have the day off on Monday, so I am willing to cancel class for you to have a nice holiday with your family. Anyway, let's start the class now. So the main three classes of cell surface receptors we're going to be focusing on today are going to be the ion channel link receptors, of which you already are quite well aware of the acetylcholine receptor, for example. And we're going to look at two other types, the G protein link receptors and the enzyme-linked receptors. So let's take a look at these and how they work and understand how they help the cell transduce signal. Now, as you are aware, the ion channel link receptors are able to open up a channel when they bind to a ligand. The best example that we cover in class is the acetylcholine receptor. And the acetylcholine receptor, as you know, is able to open up the channel for sodium once acetylcholine binds to the beta subunits of it, and therefore it's able to transform a chemical signal into an electrical signal. So therefore this kind of receptor works really well to be able to give the information to the cell to now initiate an action potential, for example. So that is most of the information that we're going to look at this receptor, because you're pretty familiar with how this receptor works from the lectures that we have learned about the initiation of action potential in neurons and how that also happens in muscle cells. Like I mentioned to you, this kind of channel is able to take a neurotransmitter, bind to it, and convert that chemical signal into an action potential that leads the cell to a particular phenotype. So as you are aware, in the neuron, when the neuron is receiving an action potential message, the action potential will travel across the axon and reach the nerve terminal. At the presynaptic nerve terminal, you're going to have voltage-gated calcium channel that is normally closed, but when the voltage-gated channel opens as the electrical signals reach it, it's going to allow for the input of calcium inside the cell. So that influx of calcium inside the cell is going to allow for the fusion of the neurotransmitter containing vesicles, releasing that neurotransmitter to the postsynaptic cleft. In the postsynaptic cleft, it's where you have the ligand-link ion channels. So in this case, for example, the neurotransmitter could be acetylcholine receptor, it could be glycine, it could be any kind of different kind of neurotransmitters, can then bind to the receptor. When it binds to the receptor in the postsynaptic cell, that receptor channel will open up, allowing ions to flow inside the cell, and those ions are then create a membrane potential, which is the signal. So as I mentioned, it's a way to convert a chemical signal into an electrical signal. Let me give you an example of how important, for example, is calcium when as a secondary message in signal transduction with the following movie.
In this experiment, glial cells from the rat brain are grown in cell culture. Calcium concentrations are visualized with a fluorescent dye that becomes brighter when calcium ions are present. In the presence of small amounts of a neurotransmitter, individual cells light up randomly as ion channels open up and allow calcium ions to enter the cell. Occasionally, calcium waves are transmitted to adjacent cells through gap junctions at regions where the cells contact each other.
That was pretty awesome. I love the way that the cells light up when calcium concentration increases in the cell. Now, that is just an example to show you how concentrations of ions like calcium can increase and therefore, as a secondary message, affect cell function. We're going to look at a little bit more of that in the future. Now, let's take a look at our second kind of proteins, and these are going to be G protein link receptors. or GPCR, as G-protein coupled receptors. Now, this is a very large family of proteins, and most of them contains a particular structure. They have 7 transmembrane regions, meaning that a single polypeptide is able to cross the membrane 7 different times. They are involved in hormone signaling, they are involved in chemotaxis, some of them are involved also in olfactory sensing. So these are very evolutionary conserved receptors that have amazing use both medically as well and in microbiology. Now, we're going to be learning about them in the upcoming lab that is going to look at how chemotaxis can be triggered by the utilization of a ligand called interleukin-8. And interleukin-8 binds to a chemokine receptor that is one of those 7 transmembrane domains. So what I'm going to show you is a movie about this type of receptors.
Many G-protein coupled receptors have a large extracellular ligand binding domain. When an appropriate protein ligand binds to this domain, the receptor undergoes a conformational change that is transmitted to its cytosolic regions, which now activate a trimeric GTP binding protein, or G-protein for short. As the name implies, a trimeric G-protein is composed of 3 protein subunits called alpha, beta, and gamma. Both the alpha and gamma subunits have covalently attached lipid tails that help anchor the G-protein in the plasma membrane. In the absence of a signal, the alpha subunit has a GDP bound, and the G-protein is inactive. In some cases, the inactive G-protein is associated with the inactive receptor, while in other cases, as shown here, it only binds after the receptor is activated. In either case, an activated receptor induces a conformational change in the alpha subunit, causing the GDP to dissociate. GTP, which is abundant in the cytosol, can now readily bind in place of the GDP. GTP binding causes a further conformational change in the G-protein, activating both the alpha subunit and beta-gamma complex. In some cases, as shown here, the activated alpha subunit dissociates from the activated beta-gamma complex, whereas in other cases, the 2 activated components stay together. In either case, both of the activated components can now regulate the activity of target proteins in the plasma membrane, as shown here for a GTP-bound alpha subunit. The activated target proteins then relay the signal to other components in the signaling cascade. Eventually, the alpha subunit hydrolyzes its bound GTP to GDP, which inactivates the subunit. This step is often accelerated by the binding of another protein, called a regulator of G-protein signaling, or RGS. The inactivated GDP-bound alpha subunit now reforms an inactive G-protein with a beta-gamma complex, turning off other downstream events. As long as the signaling receptor remains stimulated, it can continue to activate G-proteins. Upon prolonged stimulation, however, the receptors eventually inactivate, even if their activating ligands remain bound. In this case, a receptor kinase phosphorylates the cytosolic portions of the activated receptor. Once a receptor has been phosphorylated in this way, it binds with high affinity to an arrestin protein, which inactivates the receptor by preventing its interaction with G-proteins. Arrestins also act as adaptor proteins and recruit the phosphorylated receptors to clathrin-coated pits, from where the receptors are endocytosed, and afterwards they can either be degraded in lysosomes or activate new signaling pathways.
Okay, let's take a look at this in a little bit more detail. So now, what we have over here, it's a G-protein coupled receptor. As I mentioned, a 7 transmembrane protein, and we also have the G-protein, and downstream of that G-protein we may have an enzyme. For example, the adenyl cyclase. Now, when the signaling molecule, the cytokine, the chemokine, the hormone binds to the G-protein coupled receptor, the receptor will undergo a conformational change, and that conformational change is able to recruit the G-protein. The G-protein will become activated. Eventually, the activated G protein can move and activate an enzyme that is usually inactive at the cell membrane. This is an example of a G-binding protein signaling switch, where the GDP form of the protein is off, and whenever there is a signal that induces the exchange of GDP for GTP, now that protein with GTP is on the on state. When the GTP is hydrolyzed and the phosphate molecule exits, you have now the GDP form of the protein, which goes back off. So subsequent exchange of GDP for GTP turns the protein on, and then hydrolysis of GTP turns the protein off. Now, let's take a look at this. As the movie mentioned, what we have is an alpha subunit, that is the GDP binding subunit, and we also have a beta and a gamma subunit. Those are going to be separate. They are linked to the protein by lipids, as shown over here in the image. When the single molecule binds to them, what you're going to have is the exchange of GDP for GTP, which now activate the subunits. Both subunits, the alpha subunit can work alone now, or it can work in conjunction with the activated beta-gamma subunit. And they're going to work to determine an effector activation. Now, this is going to be the next step in the process. So number one, the ligand binding activates G proteins by changing its conformation. That is going to lead for the exchange of GDP for GTP, and then the activated version of the molecule, it's going to separate or stay together, depends on the G binding protein, and induce an effect, downstream on another component of the signaling pathway. For example, in this slide over here, what we have is the activated G subunit and the activated alpha subunit, and the activated beta-gamma subunit. Activation of the alpha subunit bound to GTP is going to allow it to interact with an enzyme, and that enzyme, without interacting with the GTP bound form of the G protein, it's inactive. When they come together, now the enzyme is able to perform its function. GTP hydrolysis is going to inactivate the G binding protein, and therefore now will revert to forming an inactive complex with the beta and gamma subunits. The cycle gets repeated by having the G protein go back to the G coupled receptor to become activated one more time. So now there are a couple of diseases that can be dysregulated by G protein coupled receptor signaling. One of them is cholera. As you know, cholera is a disease produced by the bacteria Vibrio cholerae, which produces a toxin. The cholera toxin is the one mediating the diarrheal effect that is going to lead to an exchange of electrolytes that is going to induce diarrhea. Now, cholera toxin goes intracellularly. It's able to bind to the surface of the epithelial cell and go inside the cell. And there, it's going to lock the G-alpha subunit into a GTP activated state, meaning that it will not allow it to hydrolyze GTP into GDP. This is going to be now resulting in an activation of the enzyme adenyl cyclase, which is going to produce cyclic AMP, which then is going to result in water, sodium, potassium, chlorine, and carbonate ions, which are going to be secreted into the lumen of small intestines, and that is going to lead to the diarrheal effect that is characteristic of cholera. Now, the other disease that is affected by a GPCR, it's going to be whooping cough. Whooping cough is caused by the toxin from the bacteria Bordetella pertussis. And the whooping cough, it's going to cause you to wheeze and cause a whooping sound. Anyway, When the toxin binds to the receptor in the lungs, it's going to get endocytosed. And now what it's going to do is to inactivate the G-alpha protein, locking it in a GDP active state. That is basically going to prevent the cells in the lung from being able to move the mucus. And because they're unable to move the mucus of the lung, you ended up choking on the mucus, having the horrible cough that is characteristic of whooping cough. Now, let's take a look of how the G protein coupled receptors are fine-tuned and how they can be turned off. One of the proteins that we saw in the movie was the RGS, which is regulator or G protein signaling. That molecule allows the exchange of GTP. It induces the hydrolysis of GTP into GDP, turning off the G protein. Another one is going to be the G protein receptor kinase. That is the protein that is able to then phosphorylate the cytoplasmic domain of the G protein coupled receptor, allowing now the protein arresting to come and bind to the phosphorylated G protein coupled receptor. That is going to induce the endocytosis of the receptor. Endocytosis is going to be mediated by a clathrin-coated pit. We haven't learned about this yet, but we will in the next section of the class. The thing is that the G coupled receptors, G protein coupled receptors that are at the surface of the cell will be endocytosed, removing them from the membrane where they can no longer respond to signal. And usually what happens is that they could either be degraded by destruction into the lysosome, or they could be recycled. Now, there are two different types of G proteins. GS stands for G proteins that stimulate target protein activation, and GI stands for G proteins that inhibit target protein activation. And we're going to be looking at some G proteins. They usually have the subscript S or I to indicate if they're inhibitory target activation proteins or they are stimulatory target proteins. Now, one of the things that we can see is that the G protein are able to sometimes regulate ion channels. This is one of the examples that happens in muscle cells of the heart that bind to acetylcholine. So here, the acetylcholine receptor is a different kind of receptor than the one found in skeletal muscle. It's not a ligand-bound channel. It is a 7-transmembrane receptor. So acetylcholine bound to the 7-transmembrane receptor, it's going to induce activation of the G protein, in particular the beta-gamma subunit. It's going to allow it to interact with a closed channel. That closed channel is a potassium channel that has the ligand receptor in the inside. So when the activated beta-gamma complex allows for the interaction with the channel, that opens the channel to allow potassium ions to leave the cell. That is going to help depolarize the or hyperpolarize the cell, which is going to affect the membrane. So one of the things that I would like you to do is to review how the movement of those potassium ions is going to affect the cell membrane. Think about what we just discussed about the effect of potassium leaving the membrane of a neuron, for example. So in this case, it's a heart muscle cell. Now, when the GTP-bound G protein hydrolyzes GTP, now it's bound to GDP, it's able then to associate with the beta-gamma subunit, inactivating it. And that is then allows the calcium to close. Now, one of the things that we're going to see is that some of those G protein activated membrane-bound enzymes are going to result in the production of secondary messenger molecules. One of the most important ones is going to be cyclic AMP. Other ones we're going to see are going to be able to take phospholipids and break them into two subunits. One of them inositol triphosphate or IP3, and the other one diacylglycerol or DAG. So we're going to take a look at two of those enzymes. One of them, the adenyl cyclase, which is going to catalyze the reaction of ATP being cleaved and hydrolyzed into cyclic AMP, and phospholipase C, which is going to catalyze the reaction that produces IP3 and diacylglycerol. Now, those molecules are diffusible, those secondary messengers are diffusible, and they're able to act intracellularly in other molecules within the cell. This is going to help activate the amplification cascade. So as you remember from what we're discussing, we have a ligand binding to a receptor that is going to lead to cell level of activation. For example, in this case, the G protein. And eventually, when it gets to that enzyme is going to be able to start catalyzing a reaction, creating secondary messengers. The secondary message level increases dramatically as the enzyme is being active, and therefore that induces an amplification of the level of signal after ligand binding. At the end, the downstream effects resulting from the activation of those secondary messengers can have very different functions. It can affect metabolism, it can affect gene expression, and it can affect cytoskeletal proteins, therefore changing motility and shape. So we're going to take a quick look into that in a little bit more detail. So the first secondary message that I want to introduce you to is cyclic AMP. Cyclic AMP, it's generated by the enzyme adenylyl cyclase. Adenylyl cyclase will be able to take ATP and remove a pyrophosphate, that means the 2-phosphate at the very end of the ATP molecule, to generate an AMP molecule, which also then gets circularized. So the phosphate is able then to make a bond with the 3rd carbon of the ribose molecule. That is the generation of the circle. The phosphate, as you know, it's bound to the 5th carbon of the ribose molecule. And then by the effect of adenylyl cyclase is able then to catabolize the reaction, generating a cyclical molecule where the phosphate is also now having a bond with the 3rd carbon of the ribose. Eventually, for example, a cyclic AMP phosphodiesterase is able to induce a hydrophilic reaction that is going to linearize the molecule, generating AMP. A similar enzyme, guanylyl cyclase, it's able to generate cyclic GMP, which, as we mentioned earlier in the last lecture, it's required for smooth muscle relaxation. So one of the things that we'll see is how cyclic AMP is now used by the cell to generate downstream effects. So the levels of cyclic AMP are able to rise rapidly because the enzyme adenylyl cyclase is able to catalyze the reaction very quickly, increasing the concentration levels of cyclic AMP dramatically. So in here, what we have is a cell that is exposed to the neurotransmitter serotonin. Within 20 seconds, the concentration of adenylyl cyclase, as shown over here on the right, has increased dramatically, going from low concentrations, shown here in the left in blue, to really high concentrations, shown in the right in red. So how does this work? Let's take our enzyme. In this case, this is going to be a GTP, a G-coupled receptor protein that is going to bind the enzyme adrenaline. When adrenaline binds to the receptor, it's going to activate the adrenergic receptor. That activation is going to recruit the G-coupled protein, which is going to become active. So the G protein will now have GTP bound to it, and that is going to be associated with the adenylyl cyclase, activating the enzyme. When adenylyl cyclase is activated, it's able to take ATP and generate cyclic AMP. Now, the interesting thing is that cyclic AMP, as a soluble molecule, is able to diffuse through the cytoplasm and bind to an inactivated kinase called protein kinase A. Protein kinase A is kept inactive by binding to an inhibitor protein. Two inhibitor proteins bind together with two inactive protein kinase A. 4 adenylyl cyclase will bind to that inactive protein kinase A, remove the inactivating proteins, and now you result in having an active protein kinase A. That active protein kinase A can travel inside the nucleus, where it's going to bind to an inactive transcription factor called CREB. It will phosphorylate that inactive transcription factor that is going to recruit other molecules to it, and now that transcription factor in the nucleus goes from being inactive to being activated. Hello, kitty. And when it's activated, it's able then to bind to target sequence in the DNA that eventually become active and become transcribed to RNA, eventually changing the protein levels in the cell. So again, the activated protein kinase A is able to move across the nuclear port to generate and phosphorylate its target molecule. One interesting thing about protein kinase A is that it is a serine-threonine kinase, meaning that it's going to add phosphate groups into the side groups of the serine and threonine amino acids. We're going to see that other molecules are going to be tyrosine kinases mediating similar responses. In this movie, we're going to see more clearly explained how cyclic AMP signaling generates and affects cell function.
Adenylyl cyclase is a membrane-bound enzyme whose catalytic domain is activated by the GTP-bound form of the stimulatory G-protein alpha subunit, or G-alpha-S for short. Activated adenylyl cyclase converts ATP to cyclic AMP, which then acts as a second messenger that relays the signal from the G-protein coupled receptor to other components in the cell. In most animal cells, cyclic AMP activates cyclic AMP-dependent protein kinase, or pKa. In the inactive state, pKa consists of a complex of 2 catalytic subunits and 2 regulatory subunits. The binding of cyclic AMP to the regulatory subunits alters their conformation and liberates the catalytic subunits, which are now active and phosphorylate specific target proteins. In some endocrine cells, for example, the activated pKa catalytic subunits enter the nucleus, where they phosphorylate a transcription factor called Krebs. Phosphorylated Krebs then recruits a Krebs binding protein. This complex activates transcription after binding to specific regulatory regions that are present in the promoters of appropriate target genes.
So, as that movie illustrated, what we have is a secondary messenger that is able then to activate other molecules which can help then, for example, turn gene expression on. Now I'm going to show you a different kind of enzyme that is going to mediate a different reaction, and this is phospholipase C. So phospholipase C, it is a protein, oftentimes membrane bound, but not always. It could be actually recruited to the membrane, that binds to the phosphoinositol 4,5-bisphosphate, or better known and easier to remember, PIP2. So the PI4,5-bisphosphate, or PIP2, it is an inositol, a phosphoinositol phospholipid. It has 2 acyl tails, shown over here. They're bound to the glycerol molecule. It has a phosphate and an inositol sugar. But the carbons at position 4 and 5 have been phosphorylated. This particular phospholipid, it is the target of the phospholipase C enzyme. Now, by phospholipase C hydrolysis, PIP2 will be hydrolyzed into 2 products. One of them is going to be membrane bound. That is going to be the 1,2-diacyl glycerol, or DAG. It is basically the glycerol molecule maintained on the membrane by the 2 acyl groups, the fatty acids that are part of this phospholipid. The second portion of it, it's going to be the inositol portion of the phospholipid. But because phospholipase C cuts in between the glycerol molecule and the phosphate, now the remaining product is going to be an inositol sugar with 3-phosphate. Phosphate at position 1, position 4, and position 5. So that it is inositol 1,4,5,3-phosphate, also known as IP3. So, as you can remember it easy, PIP2, it's going to be hydrolyzed by phospholipase C into DAG, diacylglycerol, and IP3, inositol 1,4,5,3-phosphate. Now, let's see how that is going to help signal transduction. Let's take a receptor. That receptor is going to bind to its ligand, activating the G-protein coupled receptor. Now, once the G-coupled receptor has been activated, it's going to be able to induce a conformational change into the G protein. Now the G protein is going to bind to GTP, and it's going to have an activated subunit. The phospholipase C enzyme in this particular example, it is an inactive protein that is bound to the membrane. So when the activated G protein molecule interacts with the inactive phospholipase C, it will turn it on. The activated phospholipase C is now able to move along the membrane until it finds its substrate, the PIP2 molecule. By hydrolysis of PIP2 molecule into diacylglycerol and inositol 1,4,5,3-phosphate IP3, now you have two different secondary messengers. Diacylglycerol, being a lipid, it's going to stay bound to the membrane. IP3, being soluble, will be able to travel across the cytoplasm. So let's take a look first at what happens to diacylglycerol. Diacylglycerol is going to remain bound to the membrane, but is going to help attract a protein called protein kinase C. Protein kinase C, like protein kinase A, it's a serine threonine kinase, and they're slightly related to one another. So that is one of the first things that is going to happen. Phospholipase C cleaving PIP2, it's going to generate diacylglycerol and IP3. Diacylglycerol helps recruit protein kinase C to the membrane. Now, IP3, being diffusible, will diffuse across the cytoplasm until it reaches the ER. On the surface of the ER, you have an IP3 dependent calcium channel. So when IP3 binds to that channel, it will open the channel, allowing the release of calcium from the endoplasmic reticulum. That calcium will increase in the cytoplasm of the cell and will also bind to the protein kinase C. Now together, that protein kinase C that is bound to calcium and diacylglycerol is active, and therefore it's able to phosphorylate target molecules downstream of it. Let's take a look at that. So what are the molecules, before we go there, that can be affected by calcium? We're going to look that one of the main molecules is calmodulin. And calmodulin, it's a protein that when binds to calcium, it can activate the calmodulin kinases. So the protein targets are going to come out later when the calmodulin kinases are able to phosphorylate their targets. Now, there's ways in which we can mimic the effect of phospholipase C, and we have two pharmacological agents that allows us to do that. The first one is called PMA, of phosphate myristyl acetate. PMA is an analog of diacylglycerol, and it helps activate protein kinase C. The other pharmacological agent that we can use is called ionomycin. And ionomycin, it's a pharmacological agent that is a calcium ionophore. It is able to go inside the cell and bind to membranes, and it's able then to make the membrane permeable to calcium ions. We sort of studied another ionophore before, the DMP, and that one is able to make the membrane permeable to protons. Similarly, ionomycin is able to make the membrane permeable to calcium ions. So both the effect of PMA ionomycin can be used to synthetically activate a cell, therefore activating the cascade of protein kinase C. Now, here is a picture of the ionomycin molecule. It is a very hydrophobic molecule that is able to bind nicely to the plasma membrane or to the cytoplasmic membrane or to the endoplasmic reticular membrane, but usually the plasma membrane, allowing calcium to come in. So calcium is able to traverse the membrane, and that mimics the release of calcium that we see from the endoplasmic reticulum. Now, protein kinase C, it's a molecule that has multiple domains, and it requires calcium. So here in this image, what we have are the domains that are present in protein kinase C. Well, let's look at the 4 domains that are present that form different functional modules. The complex one, it's the one that is a domain that is able to bind to diacylglycerol. Complex 2 is the domain that is able to bind to phospholipids, helping it stay on the membrane. And that is the one that requires calcium to make it stick into the membrane. Now, there is a very long hinge region that links the complex 2 to the complex 3 and complex 4. The complex 3 and complex 4 are the catalytic kinase domains, and they're going to help perform the catalytic function of the protein kinase C, which is phosphorylation. So now, before we go there, let's remember the events that we are covering. What I want to remind you is that the activation of the G protein coupled receptor is going to turn on the G protein. The G protein is then able to activate the phospholipase C, which is present on the membrane, or in other cases, it could be recruited to the membrane. We're going to look at 2 different cases later. But the phospholipase C then is able to hydrolyze PIP2 into diacylglycerol and IP3. IP3 can diffuse to the cytoplasm and reach the endoplasmic reticulum when it will bind to an IP3-dependent calcium channel, opening the channel and flooding the cytoplasm with calcium ions. At the same time, the diacylglycerol molecule is able to recruit protein kinase C. Together with calcium, that will activate the protein kinase C and allow that molecule to now be able to be a functional kinase. Now, let's visit the next couple of molecules, and these are going to be enzyme-coupled receptors. What are the basic themes of these receptors? One is going to be that these receptors are going to bind ligands. They're also going to induce an enzymatic activity. Most of these enzyme-coupled receptors are tyrosine kinases. They're able to add phosphate to the hydroxyl group in tyrosine residues. Other ones are serine-threonine kinases. They are able to recruit adaptor proteins, and we're going to see another molecule that is going to be RAS. RAS is a different kind of GTP binding protein. This is part of the small monomeric GTP binding protein. And they are also able to activate the MAP kinase pathway. So we're going to look about and learn about all these different pathways now. When activated, the enzyme-coupled receptors are able to induce gene activation, proliferation, movement, and even differentiation. So let's take a look at how that happens. Now, one interesting property of the enzyme-coupled receptors is that they are dimers. But these dimers, on their own, they are inactive. Oftentimes, these dimers are separated but enclosed together in the membrane. So when the signal molecule comes together and links those dimers, the dimers now become active, and when they become active, they have a catalytic domain that is able to induce phosphorylation. Other ones, like the one shown here on the right, once they bind the signal molecule, they're able to bring in and recruit accessory molecules, which we tend to call them adaptor proteins. So those adaptor proteins are able then to have function. These are going to require, number one, dimerization to activate. So the monomers are inactive while the dimers are active. They are going to activate a kinase activity that is going to result in phosphorylation, and that phosphorylation will help recruit adaptor proteins with different functions. Phosphorylation, as we mentioned earlier, it's part of a protein switch mechanism. So in this case, the protein, in the absence of a phosphate, is inactive. When it's phosphorylated by a kinase, that protein now becomes active. Removal of that phosphate by a phosphatase returns that active molecule to an inactive state. So you are having a protein that is circulating back from an inactive to an active state by phosphorylation, or back into an inactive protein by the mediation of a phosphatase. Now let's take a look at this in detail. Here is a receptor tyrosine kinase, which is a class of molecule that is able to induce a tyrosine and kinase domain. One thing that I want to show you with this protein is that it has a tyrosine kinase domain, shown over here by the belly, but it looks like it's the belly of this single monomers. And when there is no ligand, that tyrosine kinase domain is inactive. So when the ligand binds to it and induces a dimerization of the receptor, that induces the kinase activity to be stimulated and therefore be turned on. The first thing that these proteins will do is that they will autophosphorylate each other. So the protein component on the right will phosphorylate tyrosine motifs on the left protein, and the protein on the left will phosphorylate tyrosine domains on the right protein. Now what you have is a receptor in which different tyrosine molecules around the cytoplasmic domain of the receptor have gone from being non-phosphorylated to being phosphorylated tyrosine. Being phosphorylated, that has changed the conformation of the protein, and that has activated the receptor tyrosine kinase. That activation is going to now allow for the recruitment of proteins that can recognize a tyrosine phospho domain, meaning a tyrosine amino acid with a phosphate attached to it. And we call those the adaptor proteins. Now, in this particular image, you have the recruitment of 3 different adaptogen proteins, one that is light blue, one that is cream color, and one that is dark blue. We're going to look in a moment at how that recruitment happens, but what I want to point out to you is that the activated receptor that has been phosphorylated is able to recruit these proteins down to initiate a signal transduction cascade. Now, those adaptor proteins shown here by the arrows are the ones who are going to use the receptor tyrosine kinase as a scaffold to assemble a signaling component and therefore initiate a signal cascade. The way in which those adaptor proteins bind is because they contain what is called an SH2 domain. And the SH2 domain comes from the term which is Sark homology 2. Now, as shown over here in the left, this image is an image of SHP2. SHP2 is a SH2 containing protein which has an active site, shown over here in light blue. That active site has a binding site which is masked by an inactive molecule. That is the SH2 domain. There's also another SH2 domain present here in this molecule. So it has 2, one over here in green and one over here in yellow. Those are going to be amino terminal SH2 domains that, in the absence of their ligand, they form a complex that completely inhibits the active site of the SHP2 molecule. Now, when a receptor is phosphorylated into tyrosine, as shown here in the right, the SH2 domain can recognize as phosphotyrosine and bind to it. The binding of the SH2 domain to the phosphotyrosine releases the active site of the adaptor molecule, allowing now that active site to engage in its catalytic activity, whatever the catalytic activity may be. Usually, these adaptor proteins are floating in the cytoplasm in an inactive form. So only when the receptor is phosphorylated at the tyrosine domains is that those inactive SH2 containing molecules are going to be recruited into the receptor and therefore now expose the active site. So let me show you an example of this. In this particular case, we have a receptor tyrosine kinase that has been engaged. It has activated the tyrosine kinase domain, and the receptor has autophosphorylated the 3 different tyrosines in each cytoplasmic tail. Now that has induced the recruitment of an adaptor protein, GRAB. GRAB is bound by its SH2 domain into the receptor tyrosine kinase. GRAB now has changed its shape, and that has allowed for a recruitment of a secondary adaptor protein called SOS. SOS is a RAS guanine exchange factor, and SOS, once it's activated, is able to help RAS, which is a monomeric GTP binding protein, to exchange the GDP that is bound to RAS into a GTP. So now that SOS is facilitated the exchange of the GDP in RAS for a GTP, RAS is an active G protein, and therefore it can perform functions to activate signals down the path. So RAS molecules, we're going to see that they are derived from the rat sarcoma phenotype, which are tumor cells that couldn't stop growing. And one of the mutated molecules found in that, it was RAS. So as you can see from this image, just like the other trimeric G-binding protein, RAS is bound to the membrane by a lipid, and therefore it can diffuse nicely across the membrane by having that lipid structure. So overactive RAS, because RAS can result in cell growth and division, that overactive RAS can lead to cancer, as it was discovered. So we have 3 different RAS genes in humans: HRAS, KRAS, and NRAS, and those are the most commonly found oncogenes in human cancers. One thing before we go is that almost all the receptor tyrosine kinase are going to activate RAS proteins, one of the 3 in humans. The RAS, as you know, is a monomer. It is structurally different from the trimeric G protein. It doesn't have the multiple units, so it's only one single protein, and therefore a lot smaller. That's why we call them the small monomeric GTP binding protein. Now, one of the pathways that is activated by RAS is the MAP, or mitogen activated protein kinase cascade. And the MAP kinase cascade is a very interesting array because each molecule activated will lead to the activation of a subsequent molecules. So what is shown here is that activated RAS is going to bind and activate what is called the MAP kinase of MKKK. So the MAP kinase, for example, RAF is one of the names that we use in immunology. When activated, it will become a kinase and is going to activate the MAP kinase. So once the MAP kinase is phosphorylated by the MAP kinase, it becomes active. So it's able then, the activated MAP kinase is able to bind to its target, which is the MAP kinase, phosphorylating it. The phosphorylated MAP kinase, in this case, one example of them is ERK. ERK is able to then phosphorylate different proteins, and it's shown over here, phosphorylating protein X, protein Y, and that is going to induce some changes in protein activity. But it's also phosphorylating transcription factors. So it's changing, for example, phosphorylation transcription factor A or B, that they're going to help with changing in gene expression. So let me repeat this because it could be quite confusing. So we have 3 kinase molecules: MAP kinase, the second one MAP kinase, and the third one MAP kinase. MAP kinase is phosphorylated by MAP kinase. MAP kinase is phosphorylated by MAP kinase. So as I mentioned, one example of it, of the MAP kinase is RAF. An example of the MAP kinase is MEK. And an example of the MAP kinase is ERK. There are multiple different MAP kinases. All these molecules initiate a phosphorylation cascade, and that phosphorylation cascade is going to lead to the phosphorylation of different target molecules, which get activated and then have different phenotypes. Another thing to mention is that those MAP kinases, all 3 of them, MAP kinase, MAP kinase, and MAP kinase, they're all serine threonine kinases, and therefore only phosphorylate in the serine threonine residues. With that, I will stop the lecture. Hopefully this will make sense to you, because especially the last part could be tricky. So I'm going to edit this, post it up for you, and I will look forward to talking to you on Wednesday. Bye-bye.